My name's Amara Mason. I am a junior at the University of Missouri studying biology and an undergraduate researcher in the Brown Microbiology Lab. In lab, we study the cell biology of the bacterial plant pathogen Agrobacterium tumefaciens. Agrobacterium tumefaciens is naturally resistant to many antibiotics. This bacterium also secretes an acidic, negatively charged exopolysaccharide called succinoglycan. The function of succinoglycan secretion is largely unknown, but studies have demonstrated roles in host invasion and acid tolerance. Here I present evidence that succinoglycan serves to protect Agrobacterium tumefaciens from three external stressors: acid stress, outer membrane stress, and antibiotic stress. In panel 1, I detailed the field's current understanding of succinoglycan and show a cartoon representation of host invasion by Agrobacterium tumefaciens. It is thought that as Agrobacterium approaches plant roots, the environment is more acidic, leading to changes in the bacterium which promote acid tolerance, including the production of succinoglycan. In panel 2, I modeled the succinoglycan biosynthesis pathway and described signaling through the ChIV-G-ChIV-I two-component system that regulates succinoglycan production. It is proposed that ChIV-G senses an environmental stress and upregulates transcription of genes involved in succinoglycan production through the transcription factor ChIV-I. The periplasmic protein XOR represses ChIV-G activation and therefore regulates the expression of genes involved in succinoglycan production, including XOA. Thus, in the absence of XOR, succinoglycan is overproduced. In contrast, the absence of XOA abolishes succinoglycan production. The third panel highlights a method we use to visualize succinoglycan production. Chalcophore white binds succinoglycan and can be visualized when illuminated under UV light using fluorescence microscopy. In wild-type cells, the polysaccharide is visualized by the blue fluorescence. However, deletion of XOA prevents succinoglycan production. In panel 4, we outline our hypothesis. Based on the properties of succinoglycan, we hypothesized that this polysaccharide may form a charge barrier and physical barrier to provide protection from external stressors, including acid, osmotic pressure, detergents, and antibiotics. In panel 5, I completed a growth curve in both neutral and acidic conditions. All of the strains grow much slower in the acid conditions, indicating that this is a suboptimal condition for A. tumefaciens growth. Delta XOA, which does not produce succinoglycan, is more sensitive than wild-type cells to acidic conditions. In contrast, XOR mutants overproduce succinoglycan and grows the best in acidic conditions. We believe that the negatively charged succinoglycan locally neutralizes the acid, promoting acid tolerance. Next, in panel 6, I used the detergent sodium diadesyl sulfate, or SDS, to test if succinoglycan provides protection from membrane disruptors. In the top panel, we monitor the optical density of the cells in the presence or absence of detergents. As you can see, in the absence, the cells remain in suspension. In contrast, very small amounts of detergent cause the cell to lyse, leading to a decrease in cell density over time. In the spot assay, we wanted to determine if cells can recover from the detergent treatment. In this assay, we spotted cells on agar plates after 5 minutes of exposure to the indicated percentage of SDS. We find that the XOR mutant is more resistant to detergents, whereas the XOA mutant is more sensitive than wild-type cells to the detergent. These findings suggest that succinoglycan protects the cells from membrane-disrupting agents, such as detergents. At the bottom center panel, I test the ability of succinoglycan to protect from antibiotic challenges. To do this, I use the disc diffusion assay. As the antibiotic diffuses into the agar, it will inhibit the growth of sensitive bacteria. The clear regions you see surrounding certain discs is the zone of inhibition. The larger the zone of inhibition, the more sensitive the bacteria are to that antibiotic. Here I show 2 representative plates. On the top left is the plate with wild type, and the plate with the XOA mutant is below. We have summarized all of our results in a graphical summary in which the size of the circle is proportional to the actual zone of inhibition. We find that succinoglycan production provides protection against antibiotics which target the bacterial cell wall. The mutants with reduced succinoglycan production, including XOA, ChIV-I, and ChIV-G, have increased sensitivity to cefoxitin and ampicillin. Overall, our results thus far suggest a direct role for succinoglycan in providing protection against external stressors. To further validate our findings, I used the spot assay to determine if succinoglycan confers resistance to another cell wall targeting antibiotic, cefzolodin. As you can see, when zero dilutions of each strain are spotted on plates without antibiotics, all strains grow equally well. Thank you for taking the time to learn more about my undergraduate research project. I would be happy to answer any questions that you may have about my work.